Hello everyone, I'm MSL Ubaidi, and this is lecture 16 or session 16 within chapter 20 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. As the chapter is about the specialized monitoring techniques, while the topic of the session is about monitoring of adaptive, basket, and umbrella trials. Introduction. Innovative trial designs such as adaptive, basket, and umbrella trials represent the cutting edge of clinical research. These methodologies enhance efficiency, flexibility, and precision, addressing some of the most pressing challenges in drug development. By accommodating real time adjustments, these trial designs allow researchers to maximize insights while minimizing unnecessary participant exposure to ineffective treatments. They play a crucial role in precision medicine where treatments are customized based on specific biomarkers or subpopulations. The key points, as we'll start with the monitoring techniques for adaptive trials. So, adaptive trials involve predefined modifications to study design based on interim analysis. So, it's a more flexible trial approach, trial method that is more flexible to adapt, to adjust, to cope within any changes or recommendations that may come up through interim analysis. And interim analysis meaning any analysis that is done for any ongoing data collection or data outcomes that is coming, yet still coming from still ongoing trials that haven't been closed or ended yet. So this dynamic flexibility improves efficiency and enhances trial outcomes. Key monitor activities here, despite of the all order, will start with ongoing adjustments. The most important point, the most important aspect in adaptive trial that we need to reflect on, which is the basic idea of flexibility or being adaptive trial. Ongoing adjustments. As adaptive trials require continuous oversight to implement changes, modifications such as patient reassignment or dose adjustments. As for example, a leukemia trial reallocates patients to the arm showing the most promise based on interim response data. Dropping futile or futile arms, ineffective or unsafe trial arms are discontinued early to conserve resources and protect participants, the patients. As for example, a cancer immunotherapy trial closed two arms midway due to lack of efficacy, refocusing on successful treatments, meaning focus and concentrate the resources and the rest of the trial on the remaining of the trial after closing the two arms. And this is as just an example. Here we have a summarizing diagram on unveiling the dynamics of adaptive trials. As here we have adaptive trials as predefined modifications, interim analysis, improved efficiency, and enhanced outcomes. Continuing with the key monitoring activities, patient stratification. Adaptive trials dynamically allocate participants based on biomarkers, progression rates, or other factors. As for example, genetic testing identified a subset of patients more likely to respond to our targeted therapy for breast cancer. Sample size recalculation. Interim analysis may require increasing the sample size to maintain statistical power. As for example, a cardiovascular trial adjusted participant numbers after detecting more variability in response than anticipated. Stopping criteria. Trials set predetermined rules for early termination based on efficacy or futility. Example, a COVID-19 vaccine trial halted early when efficacy goals were met in interim data. The key takeaway here from the key monitoring objectives or roles in the adaptive trials. As an effective monitoring in adaptive trials requires robust interim analysis, clear stopping mechanisms, and compliance frameworks to manage real-time modifications. Now, monitoring techniques for basket trials. And basket trials, um, as it is, if you can imagine the basket itself, as you drop, as kind of you drop one therapy or one medication into a group or a sum of diseases. So it is basically simply as doing a trial for one certain therapy or medication to treat uh, um, groups of diseases, more than one disease, multiple diseases or disorders. So basket trials focus on testing a single therapy across multiple diseases. However, we need to know that multiple diseases that share molecular or genetic features. So there have to be, for these multiple diseases, there have to be something in common in order to test the single therapy or medication for them, as these diseases, they have to share uh, certain molecular or genetic feature. Their value lies in their ability to generalize findings for biomarker-driven therapies across indications. So the core monitoring strategies in basket trials: patient segmentation, identifying and enrolling patients whose molecular characteristics match trial requirements ensures trial applicability. As for example, HER2-positive tumors in breast, gastric, and colorectal cancers are grouped under one basket trial. So, although different cancers, as this example might reflect the explanation easily and clearly, different cancers, colorectal cancer and gastric cancer and breast cancer. So different cancers, testing, let's say, one therapy or one medication for those cancers. However, these cancers share the same uh, um, biomarker, which is HER2 or HER2, which applies to the definition of that of sharing molecular or genetic feature. So multiple diseases, multiple cancers sharing same biomarker of HER2 here too. Real-time biomarker validation, molecular profiles must be continuously verified to ensure eligibility during trial enrollment. As for example, genomic sequences confirms the presence of CRAS or KRAS mutations before assigning participants to the trial. Before proceeding, as we have here a summarized diagram showing the shared molecular features in basket trials. As here we have the diseases, as here we have the molecular genetic features, and in common the shared biomarkers. Continuing with the monitoring strategies, core monitoring strategies, or for basket trials, interim efficacy analysis, subgroup analysis or analysis are critical to identifying low-performing indications that may merit an altered strategy. As for example, a basket trial for BRAF or BRAF mutated tumors discontinued the pancreatic cancer arm early due to poor efficacy results. Efficacy comparisons across subgroups. Consistent monitoring is required to assess response variations between disease types. Example basket trials evaluating immunotherapy found notable differences in efficacy between lung and melanoma cohorts, two different cancers and different findings for each. The key takeaway here from the core monitoring strategies or approaches for basket trials, as the basket trials rely on meticulous biomarker validation and subgroup monitoring to ensure accurate evaluation of cross-disease response. Now we will come to the umbrella trials, monitoring techniques for umbrella trials. And here for umbrella trials, we have multiple therapies, multiple medications in order to test them for treating or managing one certain disease. So, umbrella trials assess multiple therapies concurrently within one disease, but across different molecular subpopulations for that particular disease. They offer robust comparisons between targeted strategies. So, the key monitoring activities here, or strategies or techniques, subtype specific enrollment ensures that each molecular group achieves adequate enrollment for statistical analysis. Example, ROS1 positive lung cancer participants require dedicated recruitment audits to maintain subgroup balance. Molecular screening support, operationalizing central genomic laboratories or labs, facilitates efficient participant stratification, as for example, using next generation sequencing, NGS for short, hubs reduces delays in molecular profiling. And here is a summarized diagram on monitoring in umbrella trials, key monitoring activities, subpopulation trials, and targeted strategies. Continuing with the key monitoring activities in or for umbrella trials, as here we have dynamic trial arm management. Arms are added or retired based on performance metrics requiring oversight of adaptive decisions. Example, introducing immunotherapy options to replace underperforming chemotherapy, drugs in an arm targeting EGFR mutations, coordinated safety reviews, monitoring toxicity levels across diverse treatment groups ensures comprehensive patient safety, as for example, tracking differences in liver enzyme elevations between chemotherapy and targeted therapy arms. The key takeaway here, as an umbrella trials, the umbrella trials depend on seamless coordination of molecular testing, adaptive arm management, and cross-arm oversight. Advanced data analysis in innovative trials. So, analytical techniques. RBM risk-based monitoring concentrates monitoring resources on high-priority data components such as rare biomarker cohorts or inconsistent endpoints. So, forecast the risks. As for example, RBM, risk-based monitoring, identified protocol deviations in a rare disease targeted therapy umbrella trial. Predictive modeling forecasts potential outcomes. So for RBM, forecast prioritize the risks, while for predictive modeling, predicting, as in predicting, forecast potential outcomes, detect, imagine, or conclude the potential outcomes or risks based on interim data trends, based on ongoing data trends, guiding trial adaptations. As predictive models using the past, using the ongoing data trends that are which is the data coming from live from ongoing yet ongoing trials. Example, Bayesian models. As Bayesian models is kind of prediction of predictive approach, predicted recruitment bottlenecks, enabling strategic reallocation, and the a characteristic of Bayesian model as using the predictive approach in monitoring by using not only ongoing data, ongoing observations, but also all past data as well. And this is only an example of predictive modeling. And here is a summarized diagram on the focused resource allocation and RBM risk-based monitoring that is used to forecast, then to prioritize risks and reallocate or direct the resources accordingly. As risk-based monitoring RBM, we have rare biomarker cohorts and inconsistent endpoints. Continuing, Bayesian adaptive design dynamically adjusts randomization probability and treatment dosing based on accumulated data. Example, allocating more patients on to outperforming immunotherapy arms in a basket trial, and accumulated data here by using the ongoing now observations and or interim observations and past all data as well to have accumulated data. Real-time genomic data integration. Genetic data are analyzed alongside clinical outcomes to assess treatment impact. As for example, linking tumor mutational burden with immune checkpoint inhibitor responses. The key takeaway here, powerful statistical and predictive tools enable swift adjustments, enhancing trial agility and accuracy. Here we have a table on challenges and solutions in monitoring as the challenge, solution, outcome. And the challenges here we have complex design logistics, interim data over interpretation, regulatory complexities, patient burden, and resource allocation imbalance. Applications and practice, as we have here, monitoring approach, real world application for each. The monitoring approaches, we have adaptive arm allocation, basket trial subgroup analysis, umbrella subtype monitoring, and predictive modeling. Next, we have tables that will summarize what we have covered through this lecture. We have table on adaptive trial techniques, process, description, example, benefits, challenges. As processes, we have sample size recalculation, dropping fertile arms, patient stratification, stopping criteria, adaptive dosage modification. We have another table on the basket trial monitoring, strategy, purpose, real world example, key benefits, implementation, challenges. As strategies, we have patient segmentation, real time biomarker validation, subgroup response monitoring, inter-disease comparisons, and cross study data alignment. Furthermore, we have a table on umbrella trial oversight, monitoring activity, description, real-time example, advantages and challenges. Monitoring activity, we have subtype-specific enrollment, molecular screening pipelines, arm-level safety monitoring, dynamic protocol modification, and cross-arm data integration. We have a table on advances in trial tech, or technology. We have feature application, real world example, advantages, limitations. As features, we have cloud data storage, wearables, Bayesian algorithms, predictive modeling platforms, and blockchain. We have table on challenges and approaches, challenge description, solution, outcome, barrier. As challenges, we have complex trial structure, interim data bias, resource allocation, regulatory complexity, and high patient burden. Application table, we have scenarios. For each scenario, key principle applied, key actions, timeline, what to watch out for. As scenarios, we have adaptive leukemia trial reallocating inefficient arms, basket trial evaluating BRAF inhibitors in diverse cancers, umbrella trial testing multiple lung cancer therapies, adaptive heart failure trial adjusting sample size, basket trial monitoring CRAs or KRAs positive treatment response, umbrella trial managing multiple treatment arms with distinct toxicity profiles, adaptive COVID-19 treatment trial halting based on success, and then with scenarios still umbrella trial using NGS for molecular screening in participants, genomic data integration for umbrella trial subgroups, and adaptive breast cancer trial adjusting participant stratification. Next, we will revise what we have covered through this lecture through some multiple choice questions, some MCQs. Question 1, what is a defining characteristic of adaptive trials? Is it A, fixed trial size with no adjustments, B, continuous adjustments based on interim data, C, uniform treatment allocation across all arms, D, exclusive use for rare diseases? So definitely it's B, as it is flexible, able to adjust, cope, and modify easily. So it is B, continuous adjustments based on interim data. B is the answer. Adaptive trials are designed to make predefined changes such as reassigning participants or adjusting dosage based on interim data analysis, improving efficiency and responsiveness during the trial. Question 2, what is the primary focus of basket trials? Is it A, testing multiple interventions in a single disease, or B, evaluating single treatment across diseases with a shared molecular target, C, randomized assignment of patients to different therapies, or D, monitoring patient safety in early phase trials? So it is between A and B, as A is for the umbrella trials, so B will be for basket as evaluating a single treatment or therapy across diseases or syndromes or disorders that would share the same molecular target or genetic target or genetic property. So B is the answer, as basket trials assess the efficacy of one treatment in various diseases that share a common biomarker or genetic mutation, enabling precision medicine approaches. Question 3, why is real-time biomarker validation critical in basket trials? Is it A, it ensures all patients receive the exact same therapy? B, it validates the trial design after it ends? C, it confirms the enrolled patients match the molecular target throughout the trial? Or D, it monitors the overall safety of all enrolled participants? So why real-time biomarker is important in basket? Because we said in basket, as we mentioned in basket or and explained earlier in basket trial, one certain therapy or medication for a group of diseases, multiple diseases that share the same genetic or biomarker property or characteristic. And here, why is real-time biomarker validation critical in such basket trial, which is a basket trial? It's because of C, it confirms that enrolled patients match the molecular target throughout the trial. Match the molecular, having the same, sharing the same molecular target throughout the trial. So C is the answer. Real-time biomarker validation ensures that patients genuinely align with the genetic or molecular target of the trial, maintaining the relevance and accuracy of its outcomes. Question 4, how do umbrella now? The question about umbrella, how do umbrella trials differ from other innovative trial designs? As we said, it's the opposite from basket. A, they test multiple therapies within a single disease divided into molecular subgroups. B, they evaluate one treatment across various diseases. C, they only involve predefined sample sizes. Or D, they require fixed data pipelines for analysis. So between A and B, as B applies for the basket trial or basket trials, B. But A is the answer for this equation for umbrella, as A, they test multiple therapies, multiple treatments or drugs within a single disease divided into molecular subgroups. So as an umbrella, the umbrella trials focus on targeting multiple molecular subtypes of the same disease and allow the evaluation of different treatments to improve personalized approaches. Question 5, what is a common challenge of innovative trial designs like adaptive basket umbrella? What challenge they do share and it's common in between these three trials, and how can it be addressed? Is it A, rapid recruitment solved by patient engagement apps? B, complex design logistics solved by trial management systems? C, limited endpoint diversity solved by Bayesian statistics? Or uniform data entry standards solved by manual audits? So definitely, the common challenge in between these three types of trials or three methodologies of trials is the complexity. So B is the answer, as complex design logistics, and this can be solved or resolved by trial management systems. So B is the answer. These trial designs involve simultaneous arms or subgroups, making coordination challenging. Advanced trial management systems streamline processes and ensure seamless operations. And here we come to end of this lecture. Thank you very much and goodbye.